Today we are talking about hypotheses versus aims. So what is a hypothesis and what is an aim, and how the understanding of this is critical when you're looking at developing experiments and your understanding of how the scientific method works. Okay, so what is a hypothesis, what is an aim, and where do we go from there? We go into a lot more detail today. So a hypothesis is very simply an educated guess. Okay, so we can make a hypothesis about how things are related or what's causing something. So it is an educated guess about what causes a particular observation. So we wouldn't make a hypothesis about just some random thing. We would say, oh, we've seen this, and our hypothesis is that it's caused by X, Y, Z. An aim is slightly different, okay, and that's why we're doing this lesson, is to differentiate between the two. An aim is a very brief statement, okay, about what the purpose of an experiment is. So your hypothesis is what you think is causing the observation. Why is this rate of reaction getting faster at higher temperatures? Okay, that's then you think about developing your hypothesis. You then say, okay, well, I need to test this hypothesis by doing some experiments. And each experiment that I do is going to have a particular aim. Okay, so both of these are tied together in what we call the scientific method. So we're actually going to start today by talking a little bit about what is this scientific method, because then we can take things further. So the scientific method. You've probably heard people talking about the way that scientists think. And this is where this comes from. It's a particular way of approaching life and of approaching problems in a view to understanding and solving. So scientific method is a good thing to know in general as well, not just good for science. Okay, so the first thing, as a scientist, you make observations. Even if it's just about the world around you, you're making observations. Second thing, once you've observed something, you say, okay, well, I see that there's more plants of this type growing here and less type growing there. Less plants of that same type growing there. And you say, okay, well, what's the difference? What could be causing more plants on the top of the mountain and less plants in the valley? Okay, well, maybe it's something to do with soil type on the mountain versus soil type in the valley. Okay, and that process of thinking through what could be causing this observation that I've seen is where we are creating a hypothesis. So once we've got a hypothesis, now we can't just say that's the answer, because the hypothesis, remember, is an educated guess. It's not a statement. It's not a formula. It's not a theory. It's not an answer. Okay? It's something that we think could be the result. Now, in order to sort of say, do we believe this hypothesis or not, we're going to have to test it. So, next question would be to test your next point. Sorry, not your next question. Test your hypothesis through, and here's the important thing, well-designed experiments. And there it is, you know, well-designed experiments. So remember all the things we've been talking about with independent variables and dependent variables and controlled variables and controls for your experiment. This is also those aspects of making a well-designed experiment. Because if you've sort of testing, okay, let's the effect of soil type on where a plant grows, and now you have all sorts of other factors, you water one and not the other, then how can you say it's about soil type or not if you've got other things that are varied? So all of those steps about how reliable and accurate and trustworthy you want your experiment to go through, that's all part of building this well-designed experiments. And that also is where your aim comes in. So each of your experiments, each of your well-designed experiments, has a specific aim. So you might need a series of experiments to test your hypothesis. You might need only one experiment. It's going to depend on the hypothesis and the situation and how thoroughly you're testing it. Okay, but each time you develop one particular experiment, then that particular experiment has its own specific aim. Okay, so once we have now tested our hypothesis, we've gathered our data, we've got a result from our experiment, we need to now go back to our hypothesis and say, well, are we supporting you? Are we rejecting you? What is now the story? So we could refine the hypothesis. We're nearly there, but it doesn't seem quite right. So maybe we just need to tweak our hypothesis a little bit. Maybe it's a combination of soil and water that's affecting our plants. Okay, so we could refine it. We could support it. Oh no, everything is just about soil type. Our hypothesis was correct. Or it could be a rejection of the hypothesis. It's got nothing to do with soil type. There must be some other factor causing this difference in plants where they're growing on the mountain versus the valley. Something completely different, in which case we need to reject our hypothesis and start all over again from what could be the link that's causing our observation. So we're going to refine, support, or reject our hypothesis. Here's a thing to bear in mind through this. Okay, we're doing this, we're creating our hypothesis based on our understanding and the observations that we've made. We're testing the hypothesis through experiments that we've designed, okay, and then we're making our conclusion. But bear in mind that this is using the information that is currently available only. Also, you're using only the techniques that are currently available. To create and test the hypothesis. So it could be that you come up with this hypothesis and you come up and it's supporting, and then 15 years later somebody says, well, you know what, Mr. Smith was actually wrong about that. It's a different explanation for his observation. And that's something to bear in mind as you're looking at any scientific research and the development of science over time. There's a lovely story which I like is how the acacia with the bright green bark, how that is called a fever tree. That's because way back when, the fever trees, well, they always grow in areas alongside rivers, right? Okay. So people used to think that when you were under these trees, then you got malaria. So they called it a fever tree. It was quite a while before they realized that it's the mosquitoes that are around the water under those trees that are giving them malaria. So the best understanding that they had before they had that knowledge that the mosquitoes were there and it was mosquitoes carrying the malaria, they blamed the trees because the trees are bright green and they're weird, and it's when you go into areas with those trees then you get sick. So you've always got to bear in mind that hypotheses are developed within the constraints of what your understanding is. Your hypothesis for an observation is going to be very different to a 5-year-old's hypothesis, okay? And the hypothesis might even be different to a 50-year-old's hypothesis because you've got different experiences. So you're always looking for your observations, and you're looking to say, you know, what's the full scientific understanding that I have here, and where am I going with it? Okay, and then you're doing your experiments, and you're using whatever technology is available to you in order to answer that question, to carry out the aim for that experiment and to test that hypothesis. But bear in mind, this is not a hard and fast rule. That's why science is so fantastic, because it's continuously changing and evolving and updating over time. Right, so that is your scientific method. So let's talk a little bit more now about actually what a hypothesis is. So we've seen an observation. Okay, we've observed something, and now we want to say, okay, right, I want to create a hypothesis. I'm going to go for this next step with my knowledge and my understanding of the current situation. There's a few things that you want to bear in mind about how your hypothesis is constructed and the things that go into it. So, first thing, you've got to attempt to explain the observation or an event. Okay, so this is now we're predicting how things are related. So it's a prediction of how variables are related. It's also important to remember that your hypothesis must be complete. It must explain everything. Okay. It can't just explain only half of your observation. A hypothesis must explain your whole observation. Also, you can't hypothesize some long explanation which involves 6 different billion, gillion, zillion, and everything like that. No, this happens and that happens and something else happens and then maybe something else happens. Okay, it must be the simplest possible explanation. Okay, and the reason why it needs to be the simplest possible explanation is because we want to go from here to predictions. So it must be stated in such a way that predictions can be made. Okay, the next one. Those predictions that you make must be able to be tested with an experiment. A hypothesis that doesn't give us sort of experimental outcomes is a bit of a pointless hypothesis because it's just going to sit there as a hypothesis. Remember, it's forming part of this scientific process, this scientific method. So after the hypothesis, we need to do experiments. So we must have the ability to test these predictions through experiments to collect quantitative data. Remember, the qualitative data is just simply more observations. We want to move away from the observations here and get into the real guts of the explanation, in which case we need quantitative data. We need numbers. So, for example, we could say our hypothesis is that temperature increases the rate of respiration in yeast, increasing temperature. Okay, and we're saying this because we've done some baking. And we know that if we use cold water or if we use warm water, then our yeast responds differently and our bread rises differently. So our dough rises faster if we use warm water with our yeast. Okay, so we say this. But now remember, we must explain all the aspects of our observation, okay, in a way that predictions can be made so that we can test these things. Now, the thing is, if you increase your temperature, if you use boiling water to make your dough, you've got a problem because now no more yeast activity. So a more full observation set before making our hypothesis would be that increasing temperature increases rate of respiration in yeast to a maximum temperature, after which respiration rate decreases. So you see here, now we've got a whole lot more avenues for experiments for different aims, because we're looking at lower temperatures, we're looking for this maximum temperature, this optimum temperature, and we're looking then for a response after that. Okay, so both of these are hypotheses. This one is a not so good one. It's not as thorough, it's not as detailed. Okay, this one is a nice one. So you're always aiming for that best possible hypothesis to then work with when you're moving forward to your next step of designing your experiments and actually stating those aims. Okay, so now we're going to go on to what is an aim. So an aim is how you actually work on a detail of your hypothesis. It's how you build your experiment and how you know exactly what variables to do what with in your experiment. So an aim, first off, must work on a detail of the hypothesis. Remember, it's one aim for one experiment. What does the aim look like? It looks like a very brief sentence. It's just stating exactly what the aim of the experiment is. What's the point of this experiment? So for example, you could have an experiment to test the effect of temperature on the respiration rate of yeast. It is very specific. It's not a hypothesis about how the two are related. It's an aim that you're testing this. So the aim is going to look at what you will do to prove or disprove the hypothesis. When we're thinking about the aim, this is where we now want to think about what's my independent variables and what's my dependent variables, because it's who am I sitting and who am I measuring to see the data, to collect that quantitative data that's going to help me answer my question that is set in my aim, that's going to then feed back into my hypothesis. So your independent variable is going to be the one who you think is responsible for the observation. So if we're thinking about temperature and yeast again, okay, we think that the temperature is responsible, so therefore our independent variable is going to be temperature. Our dependent variable this is the one, remember, that's responding to the independent variable. It's the one we're going to measure. So we think that respiration rate depends on temperature. So temperature is our independent variable, and some way of measuring that respiration rate is going to be our dependent variable. Okay, because it's the one that's responding, it's the one that's going to show us what's going on. So hypothesis, how we think they are related. Aim, how specifically are we going to test what we think? So let's now take it all back to wrap up again. Returning to our hypotheses, we've done our experiments. We get our results, right? And we find that our results can support our hypothesis, but they can't prove it. Okay, we can have indirect evidence and we can have direct evidence in support of our hypothesis. It suggests we're right, but remember, we're working in those constraints of our understanding and our knowledge base, and within the constraints of the technology that's available. So we can't say this is the answer. We can say it is definitely in support of. This hypothesis is a good one. It's actually easier to do the opposite. It's easier to disprove a hypothesis. To say, well, actually, that's not true. That's not right. They are not related that way. Because there we can have a direct evidence, a direct experiment result that says, those two are not linked in that way. So what do you do in that case? What do you do if your experiment comes back and it doesn't support your hypothesis? Okay, so we don't have support for our hypothesis, and neither indirect nor direct evidence supporting the hypothesis. Well, we have to go back to that hypothesis. We're either going to abandon it or we're going to refine it. So if experiment does not support your hypothesis, then you are either going to abandon the hypothesis and start from scratch all over again, or you're going to refine the hypothesis. So maybe during your experiment, you noticed something that was a little bit wonky, and maybe that could actually lead into how this experiment could be helping you to understand your hypothesis better, and how you could then improve that hypothesis to make it a better representation. And that also can tie back to our previous hypotheses of the yeast and the respiration rate. Remember that first one we weren't so happy with, where it just said temperature increases respiration rate. So if we had worked with that hypothesis and we'd found that these really high temperatures don't increase respiration rates, but it does at lower temperatures, that's the exact thing we're talking about here, where we then go back and we refine it and say, okay, so it's not exactly what we thought, but how can we refine it? And that's when we would come to that second hypothesis, which we're happy with, where temperature increases and then respiration rate increases up to a maximum temperature, and then it decreases. The other thing to bear in mind for all of this is that repetition is important. So the more that an experiment can be repeated and have the same results and support your hypothesis, then the better understanding that you have of whether that hypothesis is supported. So if you're in your lab and somebody else in their lab and someone else in another lab on the other side of the world are all getting the same results, then it's a pretty decent hypothesis. So the questions come through of what would a proven hypothesis look like? And there is, in a way, no such thing. It's just we have more and more and more support for it, then we trust that hypothesis. And then we can develop that further into theories and understandings. So the idea here is that we're starting from the beginning when we don't know what the link is. We just have this observation and we're going to build then our hypothesis for understanding. If you then have a million different experiments that support your hypothesis, then we start to talk more about proving a hypothesis. But the number of different experiments you would have to do to really prove that this is what's going on and is not something else that's adding in, not something else that's affecting, then it's a long way and a long lot of work that we're going to have to do that. Okay, so hopefully you've now got a good idea of hypothesis versus aims and the idea of what they sort of represent within that process, that you start off with the hypothesis of how you're explaining your observations, and then the aims are the sort of intention of that specific experiment to test your hypothesis.